Good morning, Suzuki Science, grade 11. So this was the additional presentation I had promised you on Monday. I knew there's a ton of information, and so one of the reasons I did not release it on Tuesday, Wednesday, Thursday is because I did not want to overwhelm you. But this is completely a bonus for those of you totally interested in the wonderful world of living organisms, especially fungi, protists, and, well, I'm focusing on animals. So first I want to talk about the some of the questions you guys did on the BioLeague competition yesterday. There were certain questions that were done very poorly where, more than half the class got those answers incorrect. So I wanted to take a moment to just go over those questions to make sure that you're okay. One of them was, this is an example of an, so in class, we would have looked at amoeba, paramecium, euglena, slime molds. We would have seen them, explored them. You would have been very familiar with them. So it's understandable that it would just be another slide, another picture, and you wouldn't notice it as well. But this is something we will talk about in grade 12 bio. We'll talk about white blood cells and this kind of movement. So amoebas are, a little bit more important to us because of their evolutionary history and their similarity to some of our cells. So the amoeba is what you saw in that picture, with that very distinctive blob-like pseudopod motion. Right? And there was a video in the PowerPoint that allows you to see an amoeba eating a paramecium, I believe. Another question that was done poorly, fungi are like animals because they are heterotrophic. So these are good questions for us to ask, because what they're doing is they're asking us, or asking you, to compare the different kingdoms. Do you understand the general characteristics of protists? Well, that's everything that's not one of the other four. Bacteria, animals, plants, and fungi. If you can, then you understand the majority of what we really want to drive home this week in your taxonomic differentiation. So fungi are like animals because they are heterotrophic. Plants are autotrophic. Fungi and animals must absorb their food from other sources. Now this one, so it was a little bit tricky because I said it, but I never wrote it. And so, of course, you're not going to think it's important, but I did emphasize it when I was talking about it. So symmetry, your animals, the very simplest ones, your sponges, do not have any symmetry. They're asymmetrical. Then you get into your cnidarians, which have that radial symmetry, and you see that in the echinoderms as well. And then most of the animals are bilaterally symmetrical. So that means they have mirror halves, and there's an axis down the middle that can be used as kind of the mirror plane, and then the left and right are identical. And when you get a bilaterally symmetrical animal, the sensory pieces, so the eyes, the ears, the smell, the number of senses are all in the brain, are all at one spot, the anterior part of the body. And that focus of all the senses at one end of the body is known as cephalization. So that is a major evolutionary milestone associated with bilateral symmetry, which makes these animals hunters. Right? So if you'd watched some of the Shape of Life videos, you would have seen the planaria as the first hunter going around hunting food. The other question that was done poorly, and I specifically avoided putting the slide in because, again, you already had 120 slides, so I had to pick and choose, but you should know the different parts of the body. So when you take a look at an animal that is bilaterally symmetrical, especially an animal like a chordate or a vertebrate, then you should know these four terms. So dorsal refers to the top. Right? So if you're on four legs, don't think humans, because humans we walk bipedal on two legs, so it's a little weird. But four legs, then think of the spine being the dorsal surface. And then the belly, and then the belly would be the ventral, so down here below the plane. The anterior is the front, so that's where the head is. And the posterior is the rear, where the bum is. So those are the four regions of the body. Okay. And next is the protostome. So this is just a different word. It's a different language. So it's understandable that you get confused with this one here too. So protos is Latin for first, and stoma, which we saw in grade 10 with stomata, is mouth. So protos, stome means first mouth. So protostomes, the first opening during this embryological development after the blastula, is the mouth. In a deuterostome, deutero means second, stoma means mouth. So the second opening is the mouth. So that was the relationship there. So those were, I believe, 5 questions that were done very poorly that I deem important. So that's why I gave you this extra piece here. At this point, you can shut it off because these now represent the bonus lessons. Things that are completely fascinating that I was not able to share or talk to you about during class. Here we have a little tiny worm. So we've learned about 3 worm kingdoms. We learned about flatworms, platyhelminthes, roundworms, nematodes, and we've learned about segmented worms, the annelids. So this is a parasitic flatworm, Dicrocilium dendriticum. It grows about 10 millimeters by 2.5 millimeters, which tells you it is super, super tiny, right? We call these trematodes. That's a common name. And this particular trematode begins its life as eggs in the poop of cows and sheep. Shortly after being deposited in the eggs are in the poop. The eggs are ingested by a snail. And then inside the snail, they hatch into larvae. And so you can see going from egg to this kind of larval form. And that's a microscopic view right here. In this larval form, they burrow through the snail's gut and rest in its connective tissue where they develop into a second larval stage known as sporocysts, which you see here. Now they move into the digestive gland where they bear female sporocysts, and that produce another larval stage known as cercaria. These then travel to the snail's respiratory chamber, and from there the snail will rid itself of the parasite as these little slime balls. So slime balls will come right out of this section right here. Now, I know a slime ball doesn't sound the most appealing, but there are some animals that love these slime balls. And here we have Formica fusca, so looks like a black ant. So it will find these slime balls and will eat them. And inside that slime ball, as you know, there's a ton of these larval worms. So once in the ant's intestine, the cercaria are released from the ball and most migrate to the ant's main body cavity, the hemocoel, right? And in that spot, they transform into a 4th larval stage known as the metacercaria. Now, there's typically one evil genius metacercaria that does not join its siblings in the ant's hemocoel, basically in the stomach area. But rather, it travels to a cluster of nerve cells up here, and it takes control of the ant's action by manipulating these nerves. When night falls, the metacercaria then directs the ant to climb to the top of a blade of grass, where it stays until the morning. The ant will repeat this nightly ritual at the behest of its puppet master until it's eaten by a grazing animal. That's the cow or the sheep. So the fluke has now reached the body where it will finally mature, called the primary or definitive host. It's typically the mammal. It makes its way from the animal's small intestine to its bile duct, where it reproduces and makes eggs, the latter of which are pooped by the animal into the field to start the cycle all over again. So D. dendriticum is found throughout the world, and although it primarily affects sheep, cows, snails, ants, it has also been known to inhabit pigs, goats, alpacas, and llamas. In fact, while rare, human infections are not unknown, with flukes infesting human bile ducts. Usually, those infected suffer only mild symptoms, including bloating and diarrhea, although some suffer from the enlargement of the lining of the bile ducts, that along with the growth of fibrous tissue could cause the liver to swell and cirrhosis. Okay, so human infection is very rare, so you don't have to worry about this. All right. So there are many parasites I just described one life cycle. These are fascinating. I encourage you to take some of these and just Google them. If you're interested in learning about parasites, take a look at these. The lancet liver fluke I just talked about. There's the guinea worms, absolutely disgusting. I think it was President Carter in the States. He made his life mission after he came out of presidency to rid the world of these worms. Schistosomiasis, so blood flukes, trematodes, and schistosomes. These are snails, tadpoles, and birds. And then we have the Leucochloridium paradoxum. So all four of these are fascinating. There's some good National Geographic video links. I'll try to link them up for you so you can take a look at this world of parasites, worms, and animals, other animals. So the last part of this I'm going to talk about is back to arthropods. So you were supposed to do design an arthropod. This is the most populous of all the animal phyla. There's a lot of representation. On my Wild Wednesday on Wednesday, for those of you who came, you'll notice I did talk about some arthropods. I don't know if you recognize some of these. This is the cicada. I shouldn't tell you what this is. Can you tell me what these are? That's that really loud sound in the summertime that is, it just gives you a headache. How about this one here? Recognize that? Or how about this one? Do you recognize this? I have some others that I didn't bring up from downstairs. And I had these on Wild Wednesday. I talked about them in a bit more detail. Okay, so there's that. It's a giant beetle. On the right, my side here, I have some books about bees and what the world would be like without them. We started talking about the course with that same theme. So on the screen here, this is actually a representation of this photographer, David Lichtschwager. He made portraits of dozens of bugs in a class Insecta. So these are specific arthropods that belong to the subphylum Hexapoda, and specifically Insecta. There's millions of species. When adults, all have 6 legs, 3 body segments, and a rigid exoskeleton. Beyond that, diversity rules. 10 groups are represented here. Can you tell what they are? I'll say them and see if you can point them out. Beetle, which is Coleoptera, cockroach and termites, which are very similar, Blattoidea, butterflies and moths, Lepidoptera, flies, Diptera, the true bugs, Hemiptera, bees, wasps, and ants and sawflies, Hymenoptera, walking sticks, Phasmida, alderflies, Dobson flies, and fish flies, Megaloptera, nervewings, Neuroptera, grasshoppers, locusts, and crickets, Orthoptera. So these are all represented there. Could you find them in there in that picture? So arthropods. So I'm going to talk about one arthropod in particular. And this one arthropod I want to talk to you about is the mosquito. That's right, the mosquito. So there's a video there. Upload this PowerPoint. You guys can play the video, and it does a little 5-minute blurb from Global News. So it's a local story about the mosquito. It deserves many a story. So I'm going to give you a story about the mosquito. Okay. So there's a blurb. I'm going to go past that. You don't need me to read that. Thriving in wet conditions, mosquitoes are vectors for a variety of diseases, of which malaria is the deadliest. Before we take a deep dive into human history, let's first step back and meet our story's antagonist, the mosquito itself. Or rather, the mosquito herself. It's only the female mosquito which bites us to suck up our blood, potentially transferring a disease to us in the process. She uses the blood to develop her eggs. A few days after biting us, she'll lay about 200 of them on the surface of a stagnant body of water. It could be a pond, a swamp, a puddle, or even just a tiny pool of rainwater in a discarded can in your backyard. She doesn't need much to work with. That being said, the wetter the environment, the better it will serve as a breeding ground for the insect. That's why when you go to swamps and bogs and those really wet areas, there's so many mosquitoes flying around. Temperature also plays a crucial role in the flourishing of mosquitoes. They prefer temperature above 21 degrees Celsius, and they cannot survive below temperatures of 10. And they cannot survive above temperatures of 40, 45. As a result, in the temperate climates, they emerge only in the spring, summer, and fall. That's us. While in the tropical climates, they're there all year round. Warm, wet environments thus provide ideal conditions to mosquitoes as well as the diseases they carry. These diseases are caused by pathogens that use the mosquitoes as vectors, organisms by which they transmit themselves. There are at least 15 mosquito-borne diseases that affect human beings and derive from 3 types of pathogens: viruses, worms, and parasites. They include worms that cause elephantiasis, which causes extreme swelling of the limbs and other body parts, along with viruses that cause dengue, Zika, West Nile, yellow fever. Historically, however, the heaviest hitter has been the parasite that causes malaria. That's right. There are 5 types of malaria that affect human beings, the deadliest of which are vivax and falciparum, capable of causing 43-degree Fahrenheit Celsius fevers, seizures, and comas that can lead to death rates of up to 50%. Malaria began afflicting our pre-human ancestors 6 to 8 million years ago, and it's been plaguing us ever since. As it gets passed back and forth between humans and mosquitoes, the malaria parasite mutates multiple times during its multi-stage reproductive cycle. Because of its constant shape-shifting, it is hard for scientists to come up with an effective vaccine. But that hasn't stopped human beings from fighting back in a war that goes back thousands of years. So human beings have evolved a variety of genetic defenses against malaria. We've talked about genetics, so this is a perfect connect with our previous unit. Many of them have very strange sounding names like Duffy negativity or alpha thalassemia or favism. But these defenses have mixed blessings, which have often felt more like curses to their recipients. One of the most well-known examples provides a case in point: sickle cell anemia. The story of this trait began 8,000 years ago when the agricultural Bantu-speaking people of West Central Africa started settling along the Niger River. The area was great for cultivating yams and plantains. Unfortunately, it was also home to swarms of malaria-infected mosquitoes. The disease decimated the population, who were defenseless against it. But then they developed a genetic mutation that proved to be a game changer. The mutation caused the hemoglobin in the blood to be shaped like a sickle rather than an oval or a donut, like it normally is. The malaria parasite couldn't attach itself to this new shaped hemoglobin, thwarting its reproduction cycle. The result, with the sickle cell trait developed up to 90, the people with that sickle cell trait developed up to a 90% immunity against malaria. Unfortunately, they also developed an average lifespan of only 23 years, but that was long enough for them to reproduce and pass on the sickle cell trait, which made their children more likely to survive long enough to reproduce themselves. When the Bantu-speaking population started spreading south and east across Africa between 5000 and 1000 BCE, their immunity to malaria gave them a significant edge against the groups of malaria-ridden hunter-gatherers they encountered along the way. Those groups included the Khoisan people, who took refuge in the Cape of Good Hope on the southern coast of the continent. Some ethnicities within the Bantu linguistic group went on to develop more dominant inland societies, those of the Xhosa, Shona, and Zulu. Fast forward to 1652 CE, when the Dutch started colonizing parts of southern Africa. Just a scattering of the Khoisan groups living on the coast, the Dutch were easily able to occupy that area. But when they, and later the British, tried to expand inland, they encountered the powerful Xhosa and Zulu people, along with swarms of malaria mosquitoes, which routed their soldiers, but this was hardly the first time the mosquito had sent far-reaching ripples across human history. Mosquito-borne malaria played a pivotal role in both the Greco-Persian Wars and the Peloponnesian Wars. Leaving southern Africa, let's now turn to the dawn of Western civilization. We'll begin with the 5th century BCE, when there were 2 rival superpowers vying for supremacy over the Mediterranean world, the Persian Empire and the Greeks. At the time, Greece was divided into rival city-states, which were dominated by 2 coalitions, one led by Sparta and the other led by Athens. During the Greco-Persian Wars of 499 to 449 BCE, These coalitions united against the invading Persian Empire. But even with their combined military forces, they were heavily outnumbered by their powerful Persian adversaries. It looked like the burgeoning Greek civilization might be nipped in the bud before many of its history-changing innovations in science, math, and philosophy and art had a chance to take place. But Athens and Sparta ended up getting rescued by a third ally, the mosquito. As the Persians invaded Greece and laid siege to Greek towns, they had to pass through and sometimes encamp near mosquito-filled swamps. A lethal combination of malaria and dysentery killed up to 40% of the Persian forces. As a result, at the climatic battle of Plataea in 479 BCE, the Persians arrived with a very weakened army, which the Greeks were able to defeat and effectively ending the Persian invasion of Greece. Afterward, Athens and Sparta ended up turning on each other during the Peloponnesian Wars of 460 to 404 BCE. Here again, the mosquito played a major role in determining the outcome of crucial events. In 430 BCE, the Athenians were right, on the verge of victory, when a terrible plague struck their city, killing up to 100,000 inhabitants, which was 35% of the population. The cause? Most likely either malaria or a mosquito-borne disease similar to yellow fever. Later, the mosquito would come to the Spartans' rescue again. In 415 BCE, the Athenians began a 2-year siege of Syracuse, which was an ally of Sparta. It too was surrounded by mosquito-filled swamps. By 413 BCE, up to 70% of Athens' 40,000 soldiers were either dead or unfit for combat because of malaria. Those who didn't die ended up being killed, captured, or sold into slavery by their enemies. The mosquito-led defeat at Syracuse sent the Athenians into a tailspin from which they would never recover, ultimately surrendering to the Spartans in 404 BC. But it was a hollow victory, as we'll see. By the time the Spartans won the Peloponnesian War in 404 BCE, most of southern Greece lay in ruins. The devastation led by the war was further amplified by endemic malaria, which drained Greece of its population and led to farms, mines, and ports being left unattended. With southern Greece in tatters, a relatively unscathed and isolated kingdom was able to emerge and fill the power vacuum. Its name was Macedon, and it was eventually led by the man who became known as Alexander the Great. By 326 BCE, the famous Macedonian leader seemed unstoppable. First, through a mixture of war and diplomacy, he united most of Greece, with the exception of the highly weakened and marginalized Sparta. Then Alexander and his forces headed east and conquered the Persian Empire and large parts of Central Asia. The resulting territory of this empire included much of the modern-day Egypt, Syria, Jordan, Lebanon, Israel, Palestine, Turkmenistan, Uzbekistan, Tajikistan, and Afghanistan. Now, Alexander's eyes were set on India and Pakistan, but upon entering the wet and warm environs of the Indus River Valley, Alexander's army met its toughest challenger yet. You guessed it, the mosquito. Already stretched thin by years of fighting, overstretched supply lines, and increasing reliance on mercenary soldiers, Alexander's army couldn't withstand the malaria outbreaks that tore through its ranks as it traveled past the valley's mosquito-filled swamps and rivers. They ended up retreating back to the empire's territory. During the retreat, Alexander decided to take a pit stop in Babylon, where he wanted to regroup and plan his next conquest. But this was never to be. In 323 BCE, at the age of only 32, Alexander the Great suddenly died of an illness. Likely, malaria. One of the greatest conquerors in world history appears to have been defeated once again by an insect that has about the same size and weight of a grapeseed. The consequences were enormous. Before he died, Alexander was contemplating an invasion of the Far East. If it had been successful, the East and the West would have been directly linked for the first time, 1,500 years before the European traders like Marco Polo forged a connection. Instead, right after his untimely death, Alexander's mighty empire began to crumble as his generals started fighting against each other. Malaria was an important factor in the rise and fall of the Roman Empire. We think of ancient Rome, our minds think of the Colosseum, the Pantheon, and other architectural marvels. But here's the fact our more glamorizing portrayals of Rome often leave out of the picture. From ancient times to the mid-20th century, the Eternal City was surrounded by 310 square miles of marshland, known as the Pontine Marshes. And by now, you know what that means, lots of mosquitoes. and lots of malaria. As the Roman Republic grew in power and eventually became the Roman Empire, those malaria-laden mosquitoes were some of the city's greatest allies. Between 390 BCE and 429 CE, they helped ward off one invader after another. The Gauls, the Carthaginians, the Visigoths, the Huns, the Vandals. Some of the invaders, like the Gauls, were able to sack Rome, but subsequently had to retreat because their forces were so depleted by malaria. Others, like the Carthaginians, never even got that far before the mosquitoes beat them back. Without these mosquitoes, the Roman Empire might never have arisen, as the Carthaginian Empire might have destroyed it while it was still a republic. Europeans brought malaria and other diseases to the Western Hemisphere, devastating indigenous societies. You probably know that in 1492, the year that Christopher Columbus sailed west in the search of the shortcut to Asia, only to accidentally run into an island off the coast of North America. But it wasn't just Columbus and his crew who had inadvertently reached the shores of the Western Hemisphere on that fateful mistake of a journey. It was also the mosquito-borne diseases, most notably our ancient enemy, malaria. Prior to 1492, the Western Hemisphere was home to plenty of mosquitoes, but they didn't carry any diseases. When Europeans enslaved Africans landed in the Americas, they unwittingly brought disease-ridden mosquitoes with them. Those mosquitoes either displaced or infected the native mosquitoes with their pathogens. Although the non-mosquito transmitted diseases like influenza and smallpox, mosquito-borne diseases soon spread to the indigenous people of the hemisphere. Indeed, less than a year after Columbus's crew encamped on the Caribbean island of Hispaniola, the indigenous Taino people were already suffering from a terrible outbreak of malaria and influenza. As Europeans started gaining a foothold on the coast of the mainland Americas in the early 15th century, the diseases they brought with them rapidly spread inland thanks to the indigenous trading networks. The diseases acted as a powerful vanguard for the invading Europeans. In both the southeast and southwest of what is now the United States, entire indigenous communities had been destroyed or decimated by malaria long before the Europeans even stepped foot on their territories. A combination of malaria and smallpox also devastated the mighty Aztec and Incan empires in the 1520s and 30s. As a result, the Spanish conquistadors were able to conquer, in quotation marks, these advanced multi-million people strong societies with just 600 and 168 soldiers respectively. The death and destruction wrought by the European introduced diseases was apocalyptic in scale. From 1492 to 1700, the overall indigenous population of the Western Hemisphere had plummeted an estimated 95% from 100 million to 5 million, and most of the deaths were due to illness, not conquest. During the European colonization of the Americas, the mosquito played a role in establishing both slavery and revolution. As Spain, Portugal, France, and Great Britain established and expanded their colonies in the Americas during the 16th, 17th, and 18th centuries, the European colonists faced a major problem. In areas such as the Caribbean and American South, they wanted to grow massive amounts of cash crops like sugar, cocoa, coffee, tobacco, and cotton. But to do that, they needed a massive amount of labor power. At first, many turned to enslaved indigenous people and the European indentured servants. But those enslaved people and servants kept dying from malaria, which thrived in the same conditions as the crops. As a result, enslaved Africans became seen as a much more dependable and valuable source of labor. We know this history. Hailing from West Central Africa, many of them were descendants of people who developed genetic immunities to malaria thousands of years ago that made them much more likely to survive the mosquito's deadly bite. Thus, the mosquito played a major role in the emergence and proliferation of Africans as slave labor during the European colonization of the Americas. It also played a major role in the Revolutionary Wars that brought that colonization to an end. So we then see DDT, which was used to wipe out these mosquitoes that were responsible for killing so many people. And Rachel Carson suggested that this DDT is not a good thing, but people didn't listen, and DDT was sprayed anyway. The DDT killed a lot of mosquitoes, but it killed a lot of other animals, and we've stopped using it. Many of the mosquitoes were able to evolve in such a way that they were no longer susceptible to the weaker pesticides. So we couldn't use DDT, it was too strong. We couldn't use the weak ones because they were too weak. Eventually we turned to genetic ideas like CRISPR, and some of you did presentations on CRISPR. CRISPR has allowed scientists to modify the genetic basis of certain mosquitoes, and they are able to release these genetically modified mosquitoes into the population, and these mosquitoes then would, through a gene drive, produce mosquitoes that would no longer carry malaria. This is where we are today in 2020, the release of these genetically modified mosquitoes to see if they can reduce this effect. All right. When you look at the world's deadliest animal, I found this graphic from 2013. In 2013, 10 people were killed by sharks, 1,000 people by crocodiles, 2,000 by the lowly tapeworm, 2,000 by the roundworm, and schistosomiasis, which is another worm-based parasite, 10,000. Snakes, 50,000. Humans, 475,000, evidently. And the mosquito, 1,300,000. Imagine that.